Hey, Bio2 students, Mr. Yance again. We're going to try to finish up notes today. Kind of jam-packed with a lot of things, but if you have questions, virtual students email me like always, and traditional students, you can talk to me in class tomorrow. So let's jump right in and kind of wrap this up. So we've been talking about passive transport. That is diffusion. No energy is required. We always go from areas of greater to lesser. It's just going to do its thing. Okay, we don't have to do anything. There's a type of passive transport that is called facilitated, and facilitated simply means that we have to help it. Okay, so doesn't mean we still apply energy. It's still going to go from greater to lesser. It's still diffusion, but passive transport it's sort of like it's a facilitator. If you guys know what a facilitator is, like I was doing a workshop one time down in Indy, and the lady comes in and goes, "Hey, what do you need? You need a projector? You need me to run off notes? How do you want the room set up?" She was called a convention facilitator because she set up everything and got it ready. That's what passive transport is facilitating. As this is a facilitated diffusion. Okay, so it just means that molecules are still going to go down a concentration gradient from greater to lesser, but we're just going to help it go faster because some things we just need to go through a little faster, and that's the big difference. So there's a couple ways we can do that. One way is called channel proteins. So we just put a wide hallway through the membrane that allows things to pass through there faster. Okay, it's like, we got this thick membrane, things are moving through it too slow. Let's cut a channel through there. It's hydrophilic, which means it loves water. And that's going to allow water especially to pass through a little bit faster. I'll show you a picture of that in a second. It's no different than Lake Wawasee. It has all these channels cut out of it back into these subdivisions so people can get access to the lake. That's pretty much what a channel protein is, nothing more than that. Okay. Aquaporins is a channel protein, obviously in the name, that allows water to pass through. Massive amounts, especially red blood cells, be a good example of having aquaporins, which are water channel proteins. Okay, and ionic channels are what we call gated, which means it's like a floodgate. You can open and close. So we can control when something comes in. It's sort of like a drawbridge. We open it up and allow stuff through really fast. And if we don't need to, we shut it. It's gated. Okay. Nerve impulses, electrical impulses from our brain can control that. So these are basically the same principle. We're allowing, there's an aquaporin, you can see it's just a little thicker passageway. You can see it's kind of jagged a little bit. We don't want things just flowing straight through. We want to go through, but we want to control it. Ionic channels there is another example of facilitated diffusion or these channel proteins. Okay. Now kidney stones, you can see them here. What's that got to do with this? I want to show you kind of some application of these carrier proteins, and then I'll kind of go into some more detail. Is where we have a subtle change in the shape that allows things to flow through. So with carrier proteins, a little different than channel proteins. Channels, we're just cutting hallways through the cell membrane to allow stuff to pass through faster. Carrier proteins, literally something it'll change shape and allow things to go in. Okay. What we're talking about there is that picture I just had up there. So sometimes people get these nasty things called kidney stones. They're little jagged edges. They get stuck. Very, very painful. I've heard a lot of people talk about how painful these things are. And you can see this picture over here, some places where there are those little white blurbs there. What's the deal with those? Okay, so here's the deal. So we have this disease called cystinuria, which is you don't have a protein that transports. This is a carrier. It literally will grab onto cysteine, which is an amino acid, get it out of the kidneys, and then you don't get these kidney stones. All of us have that. Okay. There's a disease where people lack that enzyme, or lack that carrier protein, I should say. No, it's not always an enzyme. And they don't have something to carry it out. Now, things will pass through there, but they don't. Cysteine builds up and then forms these painful stones called kidney stones. So, we take that for granted. Carrier proteins are just latching on to something. Think of it like, let me give you a simple analogy, sort of like a tugboat. They're going to grab a hold and tug a big ship into a harbor. That's what carrier proteins do. They go find a ship, in this case an amino acid, carry it through the channel into the location it needs to go. Okay, they help. So with facilitative, we're just talking about helping get things across the cell membrane faster. Okay, still going from greater to lesser. All that stuff's still into play. Okay, now what happens if we need to move something the opposite direction? We got to go from lesser to greater. It doesn't want to do that. Naturally, our body's not going to allow things to move the opposite direction. This is like you're out canoeing and you have to paddle upstream. It takes a lot more energy, a lot more effort. Okay. So remember with Canoe Club one time, we had a group that we had to go find. Me and my wife had to jump in a canoe and go upstream and find them. And man, you talk about wearing you out and having to exert a lot of energy. That's what happens when you have to go against the current. So we're going against the current. Okay. So active transport, here's the definition, is movement of substance against the concentration gradient going from lesser to greater. And we got to get some ATP. We got to apply some energy to do that. Okay. There's some different types of active transport we're going to look at. Let's look at this one. I got a little quick video I'll show you. And I know sometimes the audio is kind of bad with that. But we have this thing called a sodium-potassium pump. And what it does, it exchanges a sodium for a potassium across a cell membrane. Okay. So what we're doing is we're applying energy. We're forcing sodium and potassium to go against the concentration gradient and to move them out of there. So let me show you a quick video that will kind of illustrate that a little bit. Okay. So here we go.
Active transport is a type of cell transport that requires the input of energy in the form of ATP. The proteins that conduct this form of transport are often called pumps because they force molecules or ions to move from an area of low concentration to an area of high concentration. This is commonly referred to as against the concentration gradient. One of the more common examples of active transport is the sodium-potassium pump. The job of the sodium-potassium pump is to move sodium ions, or Na+, out of the cell, and potassium ions, or K+, into the cell. Let's take a closer look at how this happens. The sodium-potassium pump has binding sites for 3 sodium and 2 potassium ions. Let's follow the movement of the sodium ions first. After 3 sodium ions are positioned within the carrier protein, an ATP molecule is split, releasing phosphate. This phosphate binds to a location on the exterior of the carrier protein, causing the protein to change shape. As the protein changes shape, the 3 sodium ions are released to the other side of the membrane. Next, 2 potassium ions position themselves within the carrier protein, causing it to undergo another change in shape. In the process, the phosphate molecule is released. Once the phosphate molecule is released, the carrier protein expels the potassium ions into the interior of the cell. The carrier then resumes its initial shape, completing the cycle.
Okay, so as you can see, we are going against the concentration gradient. Sodium's going from lesser to greater, potassium's going from lesser to greater. It doesn't want to do that, so we have to apply ATP. That gives us the energy, and it changes, going back to that carrier molecule, changes the shape of it so we can force one. It goes both directions. So sodium goes out, potassium comes in. If that doesn't happen, we don't survive. So sometimes we have to make things flow opposite directions. So sodium potassium pumps, one example of active transport. Okay. Now, let's not start that again. Okay. So let's look at, let's finish things up and then we'll be done. Last slide. Woohoo! Co-transport is this simply another, these are all active, going against the concentration gradient. That's coupling a downhill diffusion with an uphill transport of a substance. Okay, so I always like to kind of use an analogy. It's like you getting a lot of speed going downhill on a bicycle. So when you go up the hill, you got momentum to carry you up. Okay, going uphill is obviously going to go against the concentration gradient. So we are getting momentum built up going downhill, so we can go uphill through a constant against the grain. And this is how we do that. We're just going to flow hydrogen in, and then we're going to do a downhill diffusion out the other direction. Okay. Then we got endocytosis and exocytosis. Try not to get these backwards. Exo is secretion of a macromolecule by the fusion of vesicles. Okay. Endo is the import of macromolecules by forming a new vesicle. So what is the difference? Endo means inside, exo means outside. So endo, kind of want to focus in on that one. I'm not going to talk much about exo. We'll get it later. Phagocytosis, which means to eat. Pino means to drink. These are two where we basically are bringing in and eating up a cell and getting rid of it. It could be food, could be bacteria, could be a useless part. That's going to take active transport. That's going to require energy to do that. Pino is the same thing. It's cellular drinking, though. So instead of eating something, we're bringing in larger quantities of fluid and getting rid of excess water or things like that. They work the same way. We have to apply energy. We're going against the concentration gradient because we want water flowing the opposite direction in this case, or food going the opposite against the concentration gradient. That's just the difference between those. What that leads me up to is this last thing, and that's getting rid of bulk quantities of substances. That's called receptor mediated. These will just be matching questions on the test. I'm not going to go in a lot of detail on any of these. You may think, man, Mr. Yance is going through these really fast. I am, because we will kind of zero in on them in some other chapters. But I do want to throw this one out there. A big health issue that we have in our society is something called arterial sclerosis. So this is going to be the anatomy teacher talking to you right here. We have these cells that, and when you get a blood test, you look for LDL, which is low density lipids, and HDL, which is high density lipids. Low density lipids are lipids that don't break down real good in our bodies, cholesterol and things like that. And we have, they travel in the blood. And these receptor proteins, if they're defective and we can't get rid of this very well, some people have a genetic disposition to this, then they start to accumulate in the body. So we should use this receptor mediated. We do have a way to grab a hold of these and get rid of them in bulk quantities. Unfortunately, we don't sometimes, and then they get built up and they get deposited in our blood vessels. And you can see this is nasty. Let me show you a picture here. Okay. And you can see this is supposed to be completely open. That's this white space is all that's there. This over here is supposed to be completely open, all these pictures. And this stuff just builds up. So, I know I went through those pretty fast, but you can see the application of them. We have to be able to use different ways of getting rid of substances. And sometimes it goes with the current, and we don't have an issue with that. But sometimes we have to make things go against the current, because if we don't, like arterial sclerosis, that gives us some health issues. And same way with kidneys, if we don't get rid of extra fluids, that can build up in our bodies. All these things they're different ways of doing the same thing. It's just very important to us in the cellular level to do these things so we can stay healthy and survive. Okay. All right. That should be it. So fill in your notes. I'll have a little review game tomorrow, a little Kahoot game, and then we'll take our test on Thursday. Okay. So, hopefully you have a good rest of the day, and we will talk to you on the flip side. So, hey, we'll see you later. Thanks. Bye.